Welcome to the Deep Dive. We're your shortcut to getting properly informed, cutting through the noise to give you the core insights on complex topics.
That's right.
Today we're tackling, well, frankly, one of the biggest and toughest questions out there, abiogenesis.
Mm-hmm.
How when did life first spark from non-living stuff? And specifically, we're zeroing in on the chemical roadblocks, the real nitty-gritty problems.
Exactly. The goal here isn't to push any simple story. It's to look at what the chemistry actually tells us, what the research shows is possible, or maybe, what seems incredibly difficult based on what we know about molecules. We're sticking purely to the chemical challenges on early Earth.
Yeah, because I think lots of us have this picture, right? Like a warm little pond, some lightning maybe.
The primordial soup idea.
Right, and then bang, life kicks off. It sounds, well, almost easy in those simplified versions.
It does. But the chemical reality, that's a whole different ballgame, far more challenging. Definitely not just a poof.
Okay, so let's dive in. Paint us a picture of early Earth, say, 4 billion years ago. What are we really talking about, chemically speaking?
Well, first off, forget any idea of a nice, clean lab setup. That's crucial.
Right.
There were no companies making pure chemicals, no bottles of reagents, no handy buffer solutions to keep the pH just right.
So what was there?
Very simple stuff.
Yeah.
Basic inorganic compounds, maybe some simple organic molecules like ammonia, methane, CO2, hydrogen sulfide, maybe some formaldehyde, water, obviously, but really basic building blocks.
And none of the tools a chemist relies on today.
Absolutely none. No pure solvents, no specialized flasks or reactors designed for specific reactions, no way to pull a vacuum, control temperature precisely, or even just, pipette a specific amount of liquid from A to B cleanly.
It sounds like trying to build, I don't know, a smartphone, but you have to start by mining the silicon and copper yourself.
Yes.
Refining it all with basic tools, no instructions, and in the middle of a storm.
That's a great analogy. You're not just assembling parts from a kit. You're inventing and creating every single piece from raw, messy materials under completely uncontrolled, harsh conditions. It's chemistry in the wild, basically.
And that wild environment must have created huge chemical problems in itself.
Oh, absolutely. Fundamental problems. Let's start with molecular degradation. Molecules, especially the more complex ones you'd need for life, tend to fall apart. They decompose.
Just on their own.
On their own, yeah, or because of the conditions, the very things you might need to make them, like heat, or certain reactive chemicals like ammonia or hydrogen sulfide that were likely around, can also tear them apart really quickly. It's a constant battle against breakdown.
So the conditions for creation could also be the conditions for destruction. A chemical catch-22.
Precisely. And that leads to something we call the time is the enemy paradox, which might seem counterintuitive.
Because people always say, oh, it had millions of years.
Right, they invoke huge timescales. But for chemistry, especially a complex synthesis outside of a living cell or a lab, more time often just means more time for things to degrade, more time for unwanted side reactions to happen, and more time for useless chemical trash to build up and contaminate everything.
Unlike a chemist who can stop a reaction and clean things up.
Exactly. A chemist purifies. They stop the reaction when they have the most product, get rid of the junk, and move to the next step. Early Earth had no mechanism for that, no purity and isolation.
So everything just stays mixed together.
A messy, intractable soup. Impurities don't just sit there, they react. They consume your starting materials, they react with your desired product, turning everything into even more complex, useless gunk. Trying to get a specific complex molecule to form and accumulate in that kind of environment is incredibly difficult.
And that leads to the mass transfer problem, right? If
Yeah.
you can't purify and you lose stuff at each step.
You've got it. Even tiny losses add up catastrophically in a multi-step process if you only get, say, 99% yield at each step, which sounds good.
Yeah, 99% sounds great.
In a lab, maybe for one step. But imagine a 10-step process. You'd lose a significant chunk. Now imagine realistic prebiotic yields, which might be, 10% or 1% or even less. After a few steps, your initial starting material, even if you started with tons, is effectively gone, vanished into side products and degradation. Nature couldn't just order more starting material from a catalog.
Or even know which pathway worked before.
Exactly. Which brings us to the fundamental point, the lack of direction.
Meeting.
Chemical reactions are, well, mindless. They just follow pathways based on energy and kinetics. There's no inherent goal driving them towards creating the specific, complex, functional molecules needed for life. Molecules don't care about becoming part of a cell. Organisms care about survival and reproduction, but molecules just react. There's no built-in selection pressure at the purely chemical level pushing towards complexity and function in the way biological evolution works.
Okay, that sets a pretty challenging stage. Now let's talk about another massive hurdle I know chemists grapple with. Chirality,
Ah, yes, chirality.
or handedness. Like our hands, right? Mirror images, but you can't stack them perfectly. A
Right.
left glove doesn't fit a right hand,
That's the perfect analogy. And the crucial thing is, life is overwhelmingly homochiral. It uses almost exclusively one hand.
for example.
Well, the amino acids that make up proteins, they're virtually all the left-handed L form. The sugars in our DNA and RNA, they're the right-handed D form. And this isn't just like a slight preference, it's essential for function.
Why is it so essential? Why can't life use a mix?
Think about enzymes or the ribosome translating RNA into protein. These are intricate molecular machines. They rely on incredibly precise shapes and alignments. If you tried to build these machines with a mix of left and right-handed parts, it'd be like trying to build an engine with mirror image bolts and threads mixed together. It just wouldn't fit or work correctly.
So the specificity is lost.
Completely. Homochirality allows for that precise alignment needed for efficient and specific biochemical reactions. We also know that cells expend energy to maintain this homochirality, which tells you how important it must be.
Has anything new come up showing why it's so important, maybe right from the start?
Yes, actually. There's fascinating work on something called chiral-induced spin selectivity, or CISS.
CISO, what's that?
It shows that the handedness of a molecule actually influences how electrons move through it. Electrons seem to travel more efficiently through chiral molecules of a specific handedness.
And why does that matter for early life?
Because efficient electron transfer is fundamental. Think about processes like respiration, moving electrons across membranes to generate energy. CISS suggests that homochirality might have been critical for these basic energy processes right from the get-go, not something that just emerged later by chance from a mixed racemic soup.
Wow, okay, so life needs homochirality. But the big question is, how do you get it? Especially on early Earth. Because normal chemistry tends to make.
A 50-50 mixture, what we call a racemic mixture. Synthesizing just one enantiomer, one-handed version is one of the hardest challenges for synthetic chemists even today, with all our knowledge and tools.
And for complex molecules.
It gets exponentially harder. A molecule might have multiple chiral centers. Just 10 centers gives you over 1,000 possible combinations 2 to the power of 10. Biological molecules can have hundreds. Controlling all of those centers to get only one specific combination out of billions or trillions, well, prebiotically. Honestly, the scientific community, looking squarely at the chemistry, has no established mechanism. We're kind of clueless on how nature solved this without biology already being present.
That really drives home the difficulty.
Yeah.
But you still hear people say things like, oh, synthesizing biomolecules is easy now, or that machines make it trivial. How does that square with these challenges?
That statement, frankly, reveals a disconnect from the actual chemical work involved, especially in a prebiotic context. Let's break it down for the main types of biomolecules.
Okay. Where should we start? Carbohydrates, sugars.
Sure. Sugars are often presented as simple repeating units, but they're incredibly complex. First, there isn't just one sugar monomer, there are many different types, and how they link together matters hugely alpha or beta linkages, connecting at different points.
How complex can it get?
Just linking 6 units of plain old D-glucose, there are over 1 trillion possible ways they can connect. And crucially, making the basic sugar monomers themselves, like ribose for RNA, in a pure homochiral D form using plausible prebiotic chemistry, that hasn't been convincingly demonstrated.
What happens in experiments?
Attempts often yield complex, messy mixtures that degrade quickly. Many famous prebiotic sugar synthesis papers actually use what's called relay synthesis, meaning they don't make the key intermediate sugar from scratch under prebiotic conditions. They buy it from a chemical supplier where it was originally sourced from living organisms, and then do the next step. It sidesteps the hardest part of the problem.
Okay, what about peptides for proteins?
Similar issues. First, the building blocks, amino acids. Experiments like Miller-Urey produce them, sure, but as racemic mixtures, that 50-50 L and D mix, life uses almost exclusively L-amino acids. Getting only the L form prebiotically is that same chirality problem again.
And linking them together.
Also tricky in water. Amino acids have both positive and negative charges. There's deuterionic, which makes forming peptide bonds a reaction that releases water difficult in an environment full of water. People talk about mineral surfaces maybe helping.
Like catalysts.
Yeah, possibly acting as catalysts to select for one chirality or promote bonding. But the evidence for this working efficiently and selectively under realistic early Earth conditions, it's speculative, not strongly supported by robust experiments yet.
And how do chemists make peptides today?
We buy purified, protected, already homochral amino acids, again, sourced from biology. We use specially designed chemicals called coupling agents, often in water-free solvents, and then do extensive purification. It's a highly engineered process, not spontaneous.
Next up, nucleotides for DNA and RNA.
Right. Well, first hurdle. You need the sugar component, which we just saw is a massive problem in itself, getting pure homochral ribose or deoxyribose.
Okay, assuming you somehow got the sugar.
Even then, making the complete nucleotide and linking it up is tough. Again, in labs today, the starting nucleotides are usually bought, often pre-protected, derived from biology. Attempts to make them from simpler precursors. They require really specific, controlled, non-prebiotic conditions.
Think what?
Things like carefully removing all oxygen, precise pH control, sometimes swinging wildly, specific wavelengths of UV light, specialized quartz tubes, stuff totally unlike early Earth. And even then, the results are often impure racemic mixtures.
What about those RNA self-replication experiments that get a lot of press? Don't they show RNA making copies of itself?
They show something interesting, but it's far from demonstrating plausible prebiotic replication. These experiments use highly engineered RNA sequences designed by humans. They need really extreme conditions, like cycling temperature from freezing up to near boiling, or big pH swings.
Not very natural.
Not at all. And they rely on lots of modern lab chemicals, surfactants, buffers. Crucially, the building blocks they feed the RNA are purchased, pure homochral nucleotides from biology. Often they need a pre-made primer strand to even get started.
And the results?
The yields are typically terrible, maybe 1% copy after weeks. Lots of errors, lots of non-functional junk RNA produced. There's no demonstration of exponential growth or a functional system emerging. Even the researchers involved acknowledge that this kind of RNA self-replication hasn't been observed happening spontaneously in nature. It's intelligent design in a lab pushing RNA to do something rudimentary.
Okay, lastly, lipids. The stuff that makes cell membranes. Are they easier?
Often portrayed as easier, but no. Again, the natural lipids we use or study, like fatty acids or glycerol, come from living things. Making them prebiotically in the right form is non-trivial. And a cell membrane isn't just a simple bag of fat. It's a highly complex, asymmetric structure needed for things like maintaining proton gradients for energy. Simple fatty acid bubbles don't do that.
And lab synthesis.
Attempts to make, say, phospholipids. They often start with purified homochral precursors bought commercially, use modern reagents in controlled conditions. Once again, it's often relay synthesis, relying on biology for the hard parts.
So across the board, it sounds like there's this huge gap between the simplified story and the messy chemical reality.
There really is a massive gap. And this hype, or perhaps oversimplification, seems to come from a few places.
Like?
While sometimes it's over-extrapolation, a researcher might achieve one specific chemical step under very controlled lab conditions, maybe making one bond type. But then it gets presented, or maybe they imply, that this somehow solves the whole pathway, or even explains the spontaneous emergence of a whole system, like metabolism. The extrapolation goes way beyond the actual data.
The language used must play a part too.
Definitely. You see the misappropriation of biological terms. Words like evolution, selection, adaptation, even metabolism get used to describe purely chemical systems where there's no replication, no inheritance, no actual biological process happening. It sounds impressive, but it can obscure the fact that the underlying chemical mechanism isn't actually established or demonstrated. Chemistry doesn't evolve or get selected in the Darwinian sense.
And you mentioned relay synthesis and human intervention multiple times.
Yes, and they're critical points. So many prebiotic simulation experiments rely on intermediates that are conveniently purchased, bypassing the extremely difficult step of how those specific pure molecules would have formed and accumulated on early Earth. And the experiments themselves involve incredible amounts of intelligent design and control, precise temperatures, pH control, adding reagents in specific sequences, purification steps, using chromatography, using specialized equipment.
Things utterly unavailable on the early Earth.
Exactly. All this human intervention, this constant guidance and purification and use of biologically sourced starting materials. When you look at it objectively, it doesn't scream, this is how life could have started spontaneously. It screams the opposite. It highlights just how incredibly difficult it is, even with all our modern intelligence and technology, to mimic even tiny steps.
It's easy to see how the public, and maybe even scientists in other fields, could get a misleading impression about how close we are to figuring this out.
Absolutely. It creates a false sense of progress sometimes. When chemists who work on synthesis look at the details, the complexity, the yield problems, the purification issues, the chirality disaster, the hurdles seem enormous and, frankly, largely unsolved from a prebiotic perspective. So let's wrap up this deep dive. We've seen the stark chemical realities facing abiogenesis. The early Earth wasn't a friendly lab. It was a harsh, messy place chemically.
Right, with constant degradation, the time is the enemy problem, no way to purify things.
Exactly. Then there's the massive unsolved problem of homochirality, getting exclusively left-handed amino acids or right-handed sugars.
Which seems essential for biological function, based on things like CISS.
Precisely. And we saw that making each class of macromolecule carbohydrates, peptides, nucleotides, lipids faces immense synthesis and assembly challenges under plausible prebiotic conditions, often masked in reports by relay synthesis and heavy human intervention.
So the overall picture from the detailed chemical literature isn't one of a clear path, is it?
Not at all. It reveals fundamental, towering obstacles. When you strip away the hype and look purely at the demonstrated chemistry, the scientific community seems, in many ways, still deeply perplexed, dare I say, clueless about how these profound chemical problems could have been overcome spontaneously on early Earth.
Which leaves us with a final thought to consider.
Yes. Given these profound, persistent, and currently unsolved chemical complexities, and given the extraordinary level of intelligent design, specific information, and continuous intervention required even in our most advanced labs to just begin to mimic the tiniest steps towards life's building blocks, what does this immense ongoing struggle truly tell us about the likely origin of the first life itself?